So I'm sure you're all very familiar with this figure. This is a figure from this classic review article, Hallmarks of Cancer, published by Bob Weinberg and Doug Hanahan in 2011. This is actually the second generation of Hallmark of Cancer. As they pointed out in this figure, genome instability is one of the hallmarks. And as you just heard from Alan and Leaf, certain cancers are associated with defects in DNA repair, and these defects in DNA repair in cancer cells render cancer cells sensitive to specific type of DNA damaging agents, and also these defects in DNA repair could render cancer cells dependent on the second DNA repair pathway, the backup pathway for survival. If you can specifically disrupt the second DNA repair pathway, you can generate synthetic lethality in cancer cells and kill them selectively. A good example of that is the use of PARP inhibitor in killing of BRCA-deficient ovarian and breast cancer cells. But today, I would like to draw your attention to a different hallmark of cancer, which could also provide an opportunity for targeted cancer therapy. To do that, I have to take you 10 years back to 2000 to look at the first generation of hallmark of cancer published by Weinberg and Hanahan, because they actually show more details in this first generation figure. As they pointed out, one of the hallmarks of cancer is limitless proliferation potential, which means that cancer cells are immortal. They can divide forever. In order for a cell to divide forever, they have to bypass senescence. This is because every time when a cell divides, their telomeres, the end of chromosomes, will become a little bit shorter. And when a cell divides many times, the telomeres will become shorter and shorter. And eventually, the shortening of telomere will trigger this process called senescence and stop cell proliferation. And for cancer cells to proliferate forever, they have to find ways to bypass senescence. They have to find ways to extend telomeres. This ability of cancer cells to bypass senescence might be a new opportunity for targeted cancer therapy. So my lab is a basic research lab. The primary research interest of my lab is trying to understand how cells detect and signal DNA damage in the genome. In particular, we're interested in this checkpoint pathway. This is not the immune checkpoint you just heard. This is the DNA damage checkpoint. So the DNA damage checkpoint pathway is a signal transduction pathway. So this pathway is activated by different forms of DNA damage. And once activated, this pathway could regulate many cellular processes, including cell cycle arrest, DNA replication, DNA repair, and so on. So collectively, this pathway is important for cells to cope with DNA damage in the genome and maintain genomic stability. So in cancer cells, in human cells, there are actually two major pathways, checkpoint pathways. One is regulated by this kinase called ATM, and the second pathway is regulated by this kinase called ATR in a complex with a protein called ATRP. So these two pathways have different DNA damage specificities. So the ATM pathway primarily responds to double-stranded DNA breaks. The ATR pathway responds not only to double-strand breaks, but also to different types of DNA replication problems. And when ATR and ATM are activated, they phosphorylate and activate downstream kinases CHEK1 and CHEK2. And together, they can phosphorylate and regulate many downstream proteins such as P53 and BRCA1 and so on. So it is important to point out that these two pathways have different DNA damage specificities. They have both overlapping and distinct functions. So my lab is particularly interested in ATR pathway because the DNA damage specificity of this pathway is much broader than the ATM pathway. So ATR is activated by many types of DNA damaging agents, chemotherapeutic drugs, and ATR is also activated in certain cancer cells by the genome instability within the cancer cells, suggesting that ATR may be a good therapeutic target in some context. So over the years, many labs, including my lab, have gradually worked out the mechanism by which the ATR pathway is activated by DNA damage. So for example, if you have a double-stranded DNA break in the genome, if the break is not repaired by end joining right away, and these breaks could be processed by exonuclease to generate single-stranded DNA at the DNA end. And single-stranded DNA at the DNA end will be recognized by this protein called RPA. So this is the predominant single-strand DNA binding protein in human cells. So RPA-coded single-strand DNA functions as a platform to recruit the ATR kinase complex and some of its critical regulators, such as the RET17 complex and RET9 complex. So once all these complexes are recruited to sites of DNA damage by RPA-coded single-strand DNA, they bring in another protein called TOP-BP1, which is a stimulator of the ATR kinase. So when TOP-BP1 is recruited to the complex, it stimulates the kinase activity and also helps the kinase to recognize some of its substrates and therefore to activate the kinase and trigger the activation of the checkpoint pathway. So in response to DNA replication problems, the pathway is also activated in a similar way. So we know that during normal DNA replication, there's a small amount of single-strand DNA in replication fork, and RPA is also a critical DNA replication protein during this process. But when replication forks encounter problems, now increased amount of single-strand DNA will be generated at replication fork, and again, it will be recognized by this protein called RPA. And RPA-coded single-strand DNA plays a critical role in recruiting the ATR kinase complex and its regulators and activators. Eventually, ATR will be activated as stressed replication forks and trigger the checkpoint signaling pathway. So the key message here is that the activation of ATR requires single-stranded DNA and this protein RPA, which recognize single-stranded DNA. So for a long time, we have been puzzled by this question. That is, how is the ATR pathway regulated at telomeres? Because if you look at telomeres are at the very end of the chromosomes. So telomeres very much look like DNA breaks. And if you look at telomeres closely, actually you can find single-stranded DNA at the very end of telomeres. So structurally, this is exactly the structure that should be activating ATR kinase. But we know that the checkpoint cannot be always activated, otherwise the cells will have problems progressing through the cell cycle. So we were wondering what's controlling ATR activation at telomeres. So this is not a question that's actually many labs have studied these questions, and in particular, the laboratory of Titia DeLange's lab at Rockefeller University and many other labs have found that this protein complex called sheltering plays an important role in protecting telomeres from being recognized as DNA breaks. So this is a complex that binds to telomere in a sequence-specific manner, because telomeres are always, they always have the same repetitive sequence, TTA-GGG. In particular, this protein, POT1, is very interesting because POT1 is actually a single-strand DNA binding protein that only recognizes telomere single-stranded DNA. So POT1 binds to telomere in a sequence-specific manner. It has been shown by several labs that POT1 actually has a critical role in preventing ATR activation at telomere. As I just showed you, RPA is also a single-strand DNA binding protein, but in contrast to POT1, RPA binds to single-strand DNA in a non-sequence specific manner. So here you can see a very unique situation at telomere. You have two single-stranded DNA binding protein. One is sequence-specific, one is non-sequence specific. One is activating ATR, and one is suppressing ATR activation. We were really curious how these two single-strand DNA binding protein are regulated at telomeres. So at the time, studies by other groups, particularly Young Carsider's group at Salk Institute, has shown that RPA actually binds to telomere in a cell cycle regulated manner. So RPA can bind to telomere during S phase when telomeres are undergoing DNA replication. But interestingly, after S phase, somehow RPA disappeared from telomeres. So now, of course, POT1 can bind to telomeres and protect telomeres from activating ATR. So we were very curious how this switch from RPA to POT1 occurs in the cell cycle regulated manner. So to address this question, so we developed an in vitro biochemical assay, try to understand how RPA deassociates from telomere in a cell cycle regulated manner. So in this experiment, so we took a piece of single-stranded DNA. So this is a single-strand DNA of telomere sequence, TTA-GGG, and we load RPA on it. So we have RPA-coded telomere single-strand DNA. And as control, we also made a piece of single-strand DNA. So now this is mutated telomere sequence. It's no longer telomere repeats. We also put RPA on it. And then we put these complexes into human cell extract and follow how long RPA can stay on DNA. As you can see, when RPA is sitting on telomere single-strand DNA, in extracts, RPA gradually comes off from single-strand DNA. But RPA stay on the mutated telomere sequence. This result suggests that there's something in human cell extracts that can displace RPA specifically from telomere single-strand DNA, but not from non-telomere sequence. So what's even more interesting is that we found that this activity is actually protein-specific. So we did a similar experiment here. So here we're looking at RPA sitting on telomere single-strand DNA. And in comparison, we also put the second protein, POT1, on telomere single-strand DNA and try the same experiment. As I showed you, RPA will gradually come off from telomere single-strand DNA, but POT1 will not come off. So this activity is displacing only RPA, but not POT1 from telomere single-stranded DNA. So to make a long story short, so we use this biochemical assay to eventually purify the factor that specifically displays RPA from telomere single-strand DNA. And this protein turns out to be HRMP-A1. So this protein was actually purified a long time ago by Tom Chek's lab to be a telomere binding protein. But its function at telomeres has been mysterious. And we showed that this protein is actually able to bind telomere single-strand DNA and displace RPA, but not POT1. And furthermore, we realized that this protein is really interesting because it is actually a DNA-RNA dual binding protein. It not only binds to single-strand telomere DNA, but also binds to a noncoding RNA that is transcribed from telomeres. And this noncoding RNA is called TERRA. So here's the model that we proposed based on our in vitro biochemical studies and also cell-based cell biology studies. So we know that during S phase, this noncoding RNA from telomeres, TERRA, is very abundant and associates with telomeres. And TERRA actually can sequester HRMP-A1 and preventing it from binding to DNA. And during this period, during S phase, RPA can bind single-strand telomere DNA very efficiently to carry out DNA replication. But interestingly, after DNA replication, in G2M phase, the level of TERRA starts to decline. So now, HRMP-A1 is released from RNA, so now it can start to bind DNA and displace RPA from single-strand telomere DNA. And during this window, G2M phase, because both RNA and DNA version, both TERRA and telomere single-strand DNA are present, so HRMP-A1 can actually shuttle between RNA and DNA in this cell cycle window. And therefore, during this period, RPA may be able to rebind telomere single-strand DNA, but because HRMP-A1 is still around, it will always get displaced right away. But for POT1, once it's bound to telomere single-strand DNA, it becomes irreversible because it could not be displaced by HRMP-A1. And through this process, POT1 will gradually take over telomere single-strand DNA. It eventually coat single-strand DNA to prevent ATR activation. So in this model, we suggest that so there's a switch from RPA to POT1 somewhere after DNA replication during G2M phase, and this transition is really driven by the up and down of this RNA called TERRA. So we decided to test this model in different cancer cell lines to see if this model is always true in different cancer cell lines. And quickly, we noticed that this model is only correct in some cancer cell lines, but not all cancer cell lines. For example, so when we tried to experiment using HeLa cells, we found that we can see TERRA sitting at telomeres in S phase cells, but in G2, TERRA disappeared. But when we look at the same thing in U2S cells, we found that TERRA actually bound to telomeres in both S phase cells and G2 cells. So for some reason, this cell cycle regulation of TERRA is compromised. It's gone in U2S cells. And when we thought about this difference, we realized that these two cell lines are actually very different because they bypass senescence in different ways. HeLa cells actually bypass senescence by activating telomerase. So this is the enzyme that can extend telomeres. In contrast, U2S cells, they do not activate telomerase, but instead they activate alternative pathway that use recombination to extend telomeres. This pathway is called ALT. So we began to suspect that the difference between these two cell lines is because they use different ways to bypass senescence. So we tested this a little further by looking at other cell lines. So here is another cell line that bypass senescence using telomerase. And again, we saw this cell cycle regulation of TERRA in this cell line. And this is another cell line that used the ALT pathway to bypass senescence. And again, we saw that TERRA stays at telomeres even after DNA replication. So we saw this correlation between how cancer cells bypass senescence and how TERRA behaves in them. So what is this ALT pathway? So ALT stands for alternative telomere lengthening. So this is a telomerase-independent mechanism to extend telomeres. So ALT pathway has been found to be active, to be used in about 10 to 15% of human cancers. So even though this is a small fraction of total human cancer, but it's actually very prevalent in specific cancer types. I will get back to that. So basically, ALT pathway, in ALT pathway, one short telomeres can use another telomere as template to extend itself through a process that's very analogous to homologous recombination. A number of DNA repair proteins have been implicated in this pathway in yeast or in human cells. This includes recombination proteins, as you heard from previous talk, RAF51. So when cancer cells use this pathway to extend telomeres, there are a number of features you can follow. The cancer cells use ALT pathway. They tend to have long and heterogeneous telomeres. Their telomeres tend to colocalize with PML bodies. This is where the ALT process takes place. And also, if you can measure telomere-specific sister chromatin exchange, a way to look at recombination rate, the all positive cells have higher telomere sister chromatin exchange, and C circles is a byproduct of telomere recombination, and all positive cells always have higher level of C circles. And I will get back to these features in a minute. So for a long time, people could not figure out why some cells activate telomerase and some other cancer cells used ALT pathway. So this began to change a few years ago when people start to sequence the cancer genome, and people finally detected association with this mutation in ATRX gene and activation of the ALT pathway. So ATRX appears to be highly mutated in several cancer types, and in some cancer types, when ATRX is lost, these cancers tend to activate the ALT pathway more frequently. So what is ATRX? So ATRX is actually a chromatin regulator. So ATRX is a family of the switch SNF family member, and it forms a complex with this protein called DAX, and together they function to incorporate a histone variant called H3.3 onto DNA. And specifically, it has been shown that ATRX is important for H3.3 deposition to telomeres. And furthermore, it has been shown by David Ellis's lab that loss of ATRX increased the level of TERRA. So this is the non-coding RNA I just mentioned that goes up and down during the cell cycle. So we started to wonder if loss of ATRX in cancer cells has some connection to the dysregulation of TERRA and the dysregulation of telomeres in cancer cells. So here we tested that idea. So we took a cell line that normally have the normal up and down of TERRA. So it's in S phase, but not in G2, sitting at telomeres. But when we knock down ATRX, so now we see TERRA foci persist into G2 after the replication. So now we can recapitulate some of the phenotypes that you only see in all cells by just knocking down ATRX. So the idea is that so when you have a lot of TERRA in G2 after DNA replication, so that will sequester hRNPA1, and that will interfere the release of RPA from telomere. And to test this possibility, so again, now we go back to the in vitro assay. So in this assay, we, again, use telomere single-strand DNA, we code it with RPA, and we put it back to extract. So now the activity in extract will release RPA and from the single-strand DNA. You can see that RPA is not very efficiently released during S phase, but in M phase, it's very efficiently released. But when we knock down ATRX, now the release of RPA becomes reduced. Again, suggesting that, so when ATRX is lost, you have high TERRA persist after replication, and consequently, you have reduced ability to release RPA. So this is another way to look at this. So here we're looking at synchronized cells progressing through the cell cycle. So if we synchronize cells in S phase, you can see some replication foci of RPA. But when cells progress into G2, RPA staining disappeared because, yeah, replication is finished. But when we knock down ATRX, so now, even when cells progress into G2 after replication, we still see a lot of RPA foci. And if you co-stain RPA with a telomere marker, TRF2, you will see that all the RPA foci are sitting at telomeres, suggesting that without ATRX, now cells have a problem releasing RPA from telomeres after DNA replication. So the experiment I showed you just now, they were done with knockdown cells. So here we're looking at a pair of isogenic cell lines. So one is telomerase positive, one is alt positive. So as you can see here, in the alt positive cell line, ATRX is not expressed, you cannot detect it, and consequently, you have a lot more TERRA, very consistent with our model. And we generate cell extracts from these cell lines, and we compare the ability of these extracts to displace RPA. And again, this is the telomerase positive cell line. RPA was displaced very effectively. This is the extracts from the alt cell line. The ability to displace RPA was significantly reduced. And again, confirming that when cells are using the alt pathway, they have reduced ability to release RPA from telomeres. So just to summarize what we learned here, we learned that loss of ATRX compromised the decline of TERRA in G2M, and loss of ATRX leads to a persistent association of RPA with telomeres after DNA replication. And we know that RPA-coded single-strand DNA is actually a key intermediate for homologous recombination. And we also know that ALT is a homologous recombination-like process. So this suggests that loss of ATRX is really giving rise to a recombinogenic structure at telomeres and therefore promoting ALT pathway, activation of the ALT pathway. But I have to point out that knockdown of ATRX or depletion of ATRX is not sufficient to activate the full ALT pathway. We suspect that this is one of the early events during the process, but additional either genetic or epigenetic events are still required to fully activate the ALT pathway. Okay, so coming back to this question, so because at the beginning I told you that single-strand DNA and RPA are important for activation of the ATR pathway, and now I show you that in all cells, telomeres have single-strand DNA, that is bound by RPA after DNA replication. So this raises the question, is ATR required for ALT? So first, we would like to know if ATR is at telomeres in all cells. As I mentioned earlier, we know that telomeres actually tend to co-localize with PML bodies in ALT-positive cells. It is also known that ATR co-localize with PML body only in ALT-positive cells. And together, this result suggests that, yes, indeed, ATR is sitting at telomeres in ALT-positive cells. So now the question is ATR required for ALT? So as I mentioned, in all positive cells, telomeres marked by TRF2 co-localize with PML body. You see this nice co-localization. So if we treat cells with ATM inhibitor, so this is the other checkpoint kinase, if we inhibit ATM, you still see this nice co-localization between telomeres and PML body, suggesting that ATM is not required for ALT. But in marked contrast, when we inhibit ATR using either siRNA or a specific inhibitor, this particular one is from Vertex, now this co-localization between telomeres and PML body is quickly disappearing. So suggesting that ATR is required to maintain this hallmark of ALT. So we also look at other hallmarks of ALT, just to make sure that ATR is important for this process. As I mentioned, telomere sister chromatin exchange is one of the hallmarks of ALT. This is a marker for telomere recombination. All cells have much higher level of sister chromatin exchange, but when we treat cells with ATR inhibitor, you can see this clear reduction in telomere recombination. Again, confirming ATR is important. In this experiment, we're looking at C-circle level. C-circle is a byproduct of telomere recombination. And again, when we treat all cells with ATR inhibitor, we see a clear reduction of C-circle levels. Again, confirming that ATR is important, functionally important for the ALT pathway. So here we wanted to conclude that ATR is a key regulator of ALT. And this raised another question. So what is the consequence of ATR inhibition in all cells? Yes, you're disrupting the pathway, but so what? So we noticed that when we treat cells with ATR inhibitor, ALT-positive cells, telomeres becomes much more fragile. Yeah, so we can actually visualize telomeres by using specific DNA probe. We can label both the leading strand, lagging strand, 2 telomeres on DNA. But when we treat cells with ATR inhibitor, these are ALT-positive cells, we saw an increased frequency of telomere loss. So telomeres becomes more fragile. In addition, we found that DNA damage started to accumulate, DNA breaks started to accumulate at at telomeres. So here we're looking at formation of 53 BP1 foci, which is a marker of DNA breaks. So in control cells, when we treat cells, when we look at 53 BP1 foci and telomere, you don't see significant colocalization. In fact, you don't see a lot 53 BP1 foci. But when we treat cells with ATR inhibitor, we see a lot more 53 BP1 foci, indicating that so you're now inducing a lot of DNA breaks in the genome, and many of the breaks actually colocalize with telomeres. So when you inhibit ATR, you're actually inducing DNA breaks at telomeres. So this is another readout. This is formation of micronuclei. Here we're comparing ALT-positive cell line and 2 non-ALT-positive cell lines. As you can see here, in the ALT-positive cell line, after ATR inhibitor treatment, you see a lot more micronuclei than the non-ALT cells. So we are seeing this selectivity. ATR inhibitor is only inducing dramatic instability in ALT-positive cells. So here, just another way to look at this. So we're looking at isogenic cell line pair. This is telomerase positive. This is ALT positive. When we treat them with ATR inhibitor, you see this marker, gamma H2X, show up specifically in the ALT-positive cells, suggesting that the ATR inhibitor is selectively inducing DNA damage in ALT-positive cancer cells. Okay, so ATR inhibition induces higher level of DNA damage in ALT-positive cancer cells and non-ALT cells. So can ATR inhibitors selectively kill ALT-positive cells? To address this, So first, we took the isogenic cell line again. The red one is the telomerase positive one, and the blue one is the alt positive one. As you can see, the alt positive cell line is more sensitive to ATR inhibitor than the telomerase positive cell line. So here, we are comparing the effects of ATR inhibitor on all positive line and a non-transformed line. So here, we're using a very low concentration of ATR inhibitor. As you can see, the non-transformed cell line proliferate just fine, a little bit slowed by ATR inhibitor, but otherwise, they proliferate just fine in the presence of ATR inhibitor. But if you look at this all positive cell line, so the entire population was wiped out by ATR inhibitor in just a few days. So there's a clear selectivity towards alt positive cell lines. As I mentioned, alt is particularly prevalent in specific cancer types. Specifically, alt is very prevalent in osteosarcoma and also in glioblastoma and also neuroendocrine pancreatic tumors. So here we would like to see if ATR inhibitor can selectively kill some of these cancer cell lines from these cancer types. So in this experiment, so we took a panel of osteosarcoma cell line and treat them with either ATM inhibitor or ATR inhibitor. So at the time we did the experiment, we did not know which cell lines are alt positive, which cell lines are non-alt positive, non-alt negative. So we just treat them with ATR inhibitor. You can see that these cell lines clearly segregate into two groups. So one group is very sensitive and the other group was relatively resistant. Interestingly, when we went back to check the alt status of these cell lines, all the cell lines in this group are telomerase positive, and all the cell lines in this group are alt positive. So clearly, so ATR inhibitor can segregate the cell lines into two groups. The alt cell lines are much more sensitive in the telomerase positive cell lines. In contrast to ATR inhibitor, the ATM inhibitor could not separate the two groups very nicely. So ATR has a specific role in alt, and it selectively kills alt positive cells. We also look at cell lines from glioma. So these are two glioma stem cell lines derived from patients. As you can see, so this cell line is a telomerase positive line, and this cell line is alt positive line. And again, the alt positive line was much more sensitive to ATR inhibitor compared to the telomerase positive line. So here, I would just like to summarize what we learned here. So there are two ways cancer cells can extend their telomeres. Of course, they can use telomerase to extend their telomeres. But in a subset of the cancer, they actually use the alt pathway to extend telomeres. And in this cancer cells, in this subset of cancer cells, if you inhibit ATR, ATR inhibitor actually does several things. So first, it will block the alt pathway. It will prevent cells from extending telomeres using this alt pathway. At the same time, it will induce or increase the fragility of telomeres. This is because telomeres are all repetitive sequence. It's actually a very difficult region for DNA replication to occur. So ATR is known to be important to stabilize replication forks. And when ATR is inhibited, you will actually have more fragility at telomeres. And in addition to this, we know that when we inhibit ATR in all cells, we not only induce DNA breaks at telomeres, we also induce DNA breaks elsewhere. We don't know how exactly that happens, but we suspect that because in the genome, there are other repetitive sequences, and ATR may be important for keeping those places stable. And when you inhibit ATR, you actually induce DNA breaks throughout the genome, and you're also blocking one of the repair pathway that specifically help to maintain telomeres. And this is why alt positive cells are highly sensitive to ATR inhibitor. I would just want to point out that so this study actually provides a unique example where you can target repair proficient cancer cell using inhibitors of checkpoint or DNA repair. Because previously, people have proposed that when cancer cells are defective in DNA repair, that creates a dependency on the second DNA repair pathway, and you can target the second DNA repair pathway and kill them. But here, we are actually looking at repair proficient cancer cells. There's no DNA repair defect here. However, these cancer cells, they are dependent on a DNA repair-like process, which is oncogenic events to survive. So in this kind of situation, if you can target the right DNA repair protein or checkpoint protein, you can selectively kill cancer cells. I think the challenge for the future research is to identify this kind of DNA repair addiction in different types of cancer cells. So it has been proposed that, yes, so if one can find a drug that can disrupt the ALT pathway, perhaps this drug could be used to treat that 10 to 15% of tumors. And we would like to suggest that ATR inhibitor is perhaps one of the first of this class of inhibitors that could be used to treat ALT-positive tumors. At the end, I would like to thank the people who did the work. Rachel Flynn is the former postdoc in my lab who did most of the work. And I would also like to thank the people who helped us in this project. And thank you for your attention.
Alan DeAndrea, beautiful work, Lee. There's been a lot of interest over the years in giving inhibitors of homologous recombination to kill an ALT-responsive cell. So you show that using the ATR inhibitor might be a selective use here, but how about like an inhibitor of, a Bloom helicase, or for instance, or something like that? Would that also kill an ALT-deficient cell?
Right. So it's not clear yet, but that idea is certainly very attractive. Yeah. But I just want to point out that, so although ALT is an HR-like process, it may not be identical to ALT. So there may be interesting differences between ALT and the real homologous recombination. Will be interesting to really understand the process of ALT and figure out what are shared and then what are differences between the two processes. Yes.
Right. So another reason this is important is as some of these ATR inhibitors work their way into the clinic, the conventional view is the best biomarker for those ATR inhibitors are things like replication stress, cyclin E amplification, things like that. This would be a completely different biomarker for the use of that ATR inhibitor.
Yeah, absolutely, yeah. So I just want to mention that those biomarkers are not mutually exclusive. Yeah, so even in all cells, you may have a second reason why cells are more sensitive to ATR inhibitor. And even in non-all cells, they could be sensitive to ATR inhibitor for a different reason, yes.
Hi, Jeff Wall. So I have two questions. So the first one is in the ALT cell where you treat with the ATR inhibitor, do you see a lot of telomere fusions and formation of dicentric chromosomes?
Right. We try to look at that, but it has been difficult because cells die very quickly. So we didn't have enough time.
What do you call quickly?
So yeah, cells die in like a day or two.
Okay, so that's time for one cell division.
Yeah.
Which is the time that it would take for a telomere fusion dicentric chromosome to kill a cell by mitotic catastrophe.
Right. So yeah, we haven't looked at that very carefully, but we follow cells through mitosis. We did see this lagging chromosome phenotype, which could be indicative of that problem. But we haven't specifically looked for that.
And you talked about intrachromosomal breaks. There are intrachromosomal telomere-like sequences.
Yes.
Is it possible that POT is binding there, or you have the same kind of competition intrachromosomally?
Exactly. That's one of the hypotheses we would love to test. Yes.
And the final thing is that you have, therapeutic index is really important. A lot of the cell lines that you talked about as being sensitive to ATR inhibition that were all positive were also P53 positive.
Right.
I noticed that.
Right.
And, one of the places where you get exquisite toxicity is in the hematopoietic system. And that's where P53 is set on a knife edge of responsiveness. So I think that in addition to studying this interesting opportunity in like fibroblasts, you really should start to try to study it in the other cell types, or even in vivo, where you're likely to get the kind of dose-limiting toxicity that could limit its utility.
Yes, that's a great point. We definitely want to try that, yes.